So last lecture we ended talking about myelination, and we were talking about how the myelination makes the appearance of a white matter versus a gray matter. So when we look at a neuron, the neuron has the dendrites, the cell body, and the axon. The axon is going to be the white matter because it's the one that has the myelin. The cell body and the dendrites are going to be the gray matter. Now when we talk about the brain and spinal cord, white matter and gray matter are going to be in different areas. So we want to take a quick look at that. So the white matter, when we talk about it, are aggregations of myelinated axons. So they're bundles of nerve, and these, remember, are tracks when we're talking about the central nervous system. The gray matter are the cell bodies, the nuclei, the dendrites, and the axon terminals, and remember, these are unmyelinated. So if we look at the spinal cord, we see that the white matter is going to be located around the periphery, and it's going to contain the ascending tracts that are going to go up to the brain and the descending tracts that are going to come down from the brain. In the spinal cord as well, we see that the gray matter is going to be located in the center, and it's going to have this butterfly or H shape that we see here in this picture. The brain is going to be opposite. When we look at the brain, the inner mass of the brain is going to be the white matter, and the outer part is going to have a thin layer of gray matter. So we see that in the spinal cord, the inside is gray, the outside is white, and in the brain, the outside is gray and the inside is white matter. All right, so now let's talk about these electrical signals that neurons are going to be able to send. Now, before we can actually look at these action potentials in more detail, we need to look at these channels that are present in the plasma membrane of these neurons. These channels are going to move ions across the plasma membrane from the concentration of being from high to low. These positive cations are going to move towards negative anions and vice versa because opposites attract. When ions move, they tend to generate these electrical currents, and this will change the membrane potential, which is what your neurons will run on. And these ions will use channels, and these channels, there's different types. There are leak channels. Leak channels are going to be randomly opened and closed throughout the neuron. We see there's chemically gated channels. These are going to be open in response to normally like a neurotransmitter, a hormone, an ion, something that actually binds chemical-wise to the protein, and it causes the protein to open. We have mechanically gated channels. These are going to open in response to physical stimuli like vibration, touch, stretch, or even pressure. And then we have the voltage gated channels. These are going to open in response to changes in those membrane potentials. So as this electrical current is generated, these are going to open up. This allows for that electrical current to continue down the axons, is where we're going to find most of these voltage gated channels. So when we look at these leak channels are randomly going to open and close, and they're going to be more permeable to potassium than they are going to be to sodium. They're going to be more potassium leak channels, and they are going to be leakier than the sodium channels. These are found in nearly all areas and types of neurons. You can see them here in the picture. Now, the gated ion channels are going to be open based on some sort of stimuli. So the chemically gated channels and the mechanically gated channels are going to open, and these are going to be found mostly on the dendrites and cell bodies. They are going to be responsible for generating some graded potentials, which we'll talk about in a moment. The voltage gated channels are going to be found on the axons. These are going to generate what we call an action potential, and they're going to be responsible for providing the excitability to muscles and nerves. So here, another overview of the four basic types. You have the leak channels. These, again, randomly open and close. They're found in nearly all areas or types of the neuron. The chemically gated channels are going to open in response to neurotransmitters, hormones, or some ions. They're going to be found on the dendrites. They're going to also be found on a lot of the sensory neurons, interneurons, and some motor neurons. We do see mechanically gated channels are going to be where the gates open in response to vibration, touch, stretch, or pressure. These are found in, like, a lot of times the auditory receptors with your ear because of sound waves moving, and also touch and pressure receptors in your skin. The voltage gated channels are going to be the ones that are going to open in response to a change in the membrane potential. These are going to be found in the axons of all types of neurons. This is what gives, again, those nerves and muscles their ability to get excited and to send action potentials from one cell to the next. And as you can see here, the chemically gated channels and the mechanically gated channels are going to be for graded potentials, whereas voltage gated are going to be for action potentials. So we see that the neuron is going to have a resting membrane potential. This membrane of a non-conducting at-rest neuron normally is positively charged on the outside of the neuron and negatively charged on the inside of the neuron. It sits at about a negative 70 millivolts. When this is the case, the membrane is considered polarized because we have a positive end, we have a negative end. We see that the membrane are more permeable to potassiums than they are sodiums. So the potassiums will leak out quicker than the sodiums will leak in. This is, again, through just simple diffusion through those leak channels. Now, the sodium and potassium pump is going to help put these back. They're going to send 3 sodiums out and 2 potassiums in, and this is, remember, an active transport which requires ATP. This is what causes that resulting positive charge on the outside of the cell. So sodium and potassium ion channels are going to leak, and this is going to move sodium and potassium into the cell, but we see that the sodium and potassium pump is going to end up creating this gradient, maintaining this resting membrane potential. So let's talk a little bit about graded potentials, where we're talking about either hyperpolarization or depolarization. Graded potentials are going to be a small deviation off of that resting membrane potential. So it's going to cause it to change a little bit off of that -70. We see that a hyperpolarized graded potential is where the inner membrane becomes even more negative. So if it started at -70, maybe it becomes -75, or maybe it gets closer to -80, but we are making it more negative. When we're talking about a depolarized graded potential, this is where the inner membrane becomes less negative. This could be where it goes up to -65 or -60. The depolarized graded potentials, as they get closer to a certain point, they are going to allow for action potential to be made. Now, this occurs again due to the opening and closing of mechanical or chemically gated channels. And a lot of times it's going to be these chemically gated channels due to neurotransmitters. There are no voltage gated channels that are part of this graded potential. When ions move through these graded channels, the membranes will either depolarize or hyperpolarize the membrane. This does depend on the charge of the ions and if they're flowing into the cell or out of the cell. So when we look at this, if a cell becomes hyperpolarized, the membrane starts to become more inhibited. But if a cell becomes depolarized, we see that it's going to cause that membrane to become more excitable. So graded potentials are going to vary in size, and this depends on the strength of the stimulus. So the greater the stimulus, the more channels that get open, whether they're mechanical or chemical, and the greater the change of the membrane that will occur. So we see here that we have a small change. We went from -70 to about -65. Then in the second stimulus, it was higher, and so we see that it was going to go up to about -60. And then the third stimulus is even more, and it's getting up closer to that -55 mark. So we see a small stimulus is going to create a small graded potential, whereas a larger stimulus is going to cause a larger graded potential. Now, the size of the graded potential is going to determine the depolarization and how excited the neuron will get. It is also going to depend on the strength of the stimulus. So we do see in this case that with graded potentials, we have a concept called summation. This is when 2 or more graded potentials are going to get added together, and this creates a greater change in the membrane potential. So if you'll notice here, we had one wave that started, but then another stimulus came in and it started again. So we see that it's a summation. We were able to add these stimuli together, a greater graded potential to occur. So when we look at this graded potentials, they're going to generate on the membrane of dendrites and the cell body, not the axons. Typically, sometimes they do happen on the axons, but it's going to remain more localized. It does not travel very far, so this is a short distance communication only with graded potentials. Now let's talk a little bit about the actual action potential. This is that impulse that the nerve then sends. An action potential is a sequence of rapidly occurring events that depolarizes the membrane, but then the membrane has to go through the process of repolarizing. So an action potential, unlike a graded potential, is going to be generated on the membrane of the axons, and this is due to the voltage-gated channels. This is going to be good for short and long distance communication, because in the brain we see it's still short distance, but if we want to send a signal from like your big toe up to your spinal cord, that could be a very long distance. When we look at action potentials, guys, they are an all or nothing principle. An action potential requires a threshold stimulus of about a -55 millivolts. Once it reaches that -55 millivolts, we see that the depolarization of the membrane is going to take place, and it's going to depolarize the whole axon. Okay, and so this is where it's at all or nothing. We get to that -55, and if we get there, we're sending the signal. If we don't get to -55, we're not. Okay, so that's kind of like the cutoff point here. So if we look here at this first particular stimuli here in the purple, the threshold was not met, so depolarization does not occur, so no action potential is sent. However, when we look at the second one, the threshold was met at the -55, so then action potential is generated. You see that it goes all the way up to a positive about 30 to 40, and then it's going to have to reset. That's that repolarization that has to occur. Now, when we are looking at these action potentials, you're going to see that there are no summations that are present. The greater the strength of the stimuli, the greater the frequency that the action potential is sent, not that it's going to be larger. If you notice, all of them go up to this, in this case, about a positive 30. That's it. They just may send it more frequently. Okay, so there's like a difference. This would be like one signal where the stimulus is not that great. So we sent the action potential that quick, but if the stimulus is great, it may send it like this. Okay, the difference was not what action was being done, because that clapping was the same no matter what. It was that it was happening more quickly. Okay, it was a higher frequency in the sense that I clapped more times in that shorter amount of time. So the stimulus intensity affects the frequency, not the size of the action potential. So again, no summation is going to occur here. So let's look at these steps of how we actually generate an action potential. First, we have to start with the resting membrane potential, which is about negative 70 millivolts. This is when the membrane is polarized. It's shown here in the purple, and the voltage-gated channels of the sodium and the potassium are closed. Okay, so nothing's really happened here. We're sitting at this resting state. In the second step, though, a stimulus has caused the membrane to depolarize, and it has reached that negative 55 millivolt threshold. Since we've met that negative 55, we see that we are going to depolarize the rest of the cell. The action potential is now going to be sent. So step 3 here is in the green. This is the depolarization phase. This is when the voltage-gated sodium channels are going to open. This allows sodium to rush in. Now remember, sodium has a positive charge, and so as it's rushing into the cytosol, we see that the inside of the membrane becomes less negative and actually starts to become positive. This causes further depolarization, which opens even more sodium-gated channels, and so this is considered a positive feedback. This influx of sodium causes depolarization to approximately a positive 30 millivolts. Step 4 is the repolarization phase. This is when voltage-gated potassium channels are going to open. Now, potassium is also positive, but in this case, instead of moving into the cell, these potassiums are going to rush out of the cell. So more positives are going to leave, which allows the cell to reset. When these potassium channels open, the sodium ones will close, and the membrane starts to repolarize. It starts to reset back down to that negative 70. Now, there is a time that there's what we call after hyperpolarizing phase. This is when the voltage-gated potassium channels remain open until the membrane potential even becomes more negative than the negative 70. It actually gets about to a negative 90 millivolts. The voltage-gated potassium channels then will close, and the resting membrane potential will return to that negative 70 with the help of the sodium-potassium pumps. So the sodium-potassium pumps will reestablish it back to that negative 70 mark. So when we look at this action potential, we see that there's always a depolarization prior to the repolarization. The whole cell is going to depolarize in order to send the action potential down the axon, and then the cell is going to repolarize to reset itself. There is what we call an absolute refractory period. This is the time when another impulse cannot be generated during the depolarization and repolarization phase. In general, guys, the frequency of nerve impulses can range anywhere between 10 or 1,000 per second. Now, there's a couple of clips here that are from Crash Course that give you an overview of how this works, along with action potentials that can be helpful to review this process. All right, so this chart's taking information that we've just covered, and it's putting it into a form where it's all on just this one slide. So when we look at graded potentials, their origin point is going to be the dendrites and the cell bodies, whereas action potentials are going to be found on the axon. The type of gated channels for graded potentials are going to be chemical and mechanical, but they're going to be voltage for action potentials. Communication distance for graded is going to be short, and for action, it's going to range from short to long. For the amplitude size of the stimulus, we see that graded potentials could vary, but action potentials remain the same. Remember, that's an all or nothing type of experience. When we look at the polarity, we see that graded potentials can either hyperpolarize or depolarize a neuron. This either inhibits the neuron or it starts to excite the neuron. Action potentials are going to always depolarize, but then they have to follow by a repolarizing stage. Graded potentials have summation, and action potentials have refractory periods. So again, this is just taking that information about graded and action potentials and putting it all here in one chart for you. All right, so when we're looking at this whole idea of conduction or propagation of these action potentials are going to arise at what we call a trigger zone. This is going to be that all or nothing state. When it hits that trigger zone, it's going to travel down the axon terminal. Now, we see that it can travel in two ways. If it is an unmyelinated axon, it's going to be continuous conduction. All right, so this means that the whole axon has to depolarize one section at a time. All right, and so this would be like if you were to walk across the room when you go heel to toe. You're going to eventually get across the room, but your steps are very small in that process because you're having to go heel to toe. So this is a slower kind of conduction. We also see that there's what we call the saltatory conduction. Saltatory means leaping in Latin, and this is going to be on myelinated axons. This is where the action potential is going to actually jump from node to node. Okay, and so this is going to be if you were going across the room, but you were leaping across the room. It's going to get there quicker. It serves the same purpose as if you were going from heel to toe to walk across the room, but you just get there faster. So the saltatory is going to allow for quicker conduction as well as allowing for it to jump between those nodes, so repolarization can also happen quicker as well. So let's take a look at the saltatory conduction a little bit closer. There is the myelin sheath that's present. Remember, this is an electrical insulator, so it blocks membrane depolarization. The nodes of Ranvier are going to contain many voltage gated channels, especially those sodium gated channels. We see that the membrane depolarizes and repolarizes very quickly at these nodes in between the myelinated groups. We see that the depolarization is going to stop at the myelin sheath. So the nerve impulse actually leaps from one node to the next, which allows for very fast travel. Also, with this type of movement, it's going to be less ATP that's required because only a small portion of the membrane gets depolarized. So the resetting with those sodium and potassium pumps requires less ATP. Now, propagation speed is also going to be related to how large the diameter of the nerve fiber is. The faster the impulse will travel, the larger it is. Think about a water hose versus a fire hose. Both of them are there to deliver water, but the fire hose is a lot bigger in diameter, and so the water is going to come out a lot quicker with a lot more force, whereas in the actual garden hose, it's not going to. So the larger the diameter, the faster the impulse. We already talked about the myelinated fibers. If they're myelinated, it's faster versus if it's unmyelinated. And also, warm nerves are going to conduct impulses faster than cooled nerves. Now, stimulus intensity, remember, is due to the frequency of the action potentials that are generated at the trigger zone, not the actual size. All right, so remember, it's going to just be where it sends a signal more often instead of making it larger in its signal. Now, signal transmission is going to occur at what we call the synapses. Now, the synapses is a functional junction that's between a neuron and another neuron, or a neuron and an effector. It's muscle or a gland. Now, these types of neurons are based on their transmission, okay? So we do see that if the neuron is before the synapses, it's called a presynaptic neuron. It's the one that's actually going to be sending the signal towards the synapses. Like you can see in this picture, we have the synapses that's highlighted here. The one before the synapses is the presynaptic neuron. The postsynaptic neuron is going to receive the signal and carry the messages away from the synapses. So it is after the synapses. That's where the post comes in. So we have presynaptic, we have postsynaptic. Now, these types of synapses can also be based on their transmissions. If a synapsis is between an axon and a dendrite, it's called an axiodendritic synapses. Okay, and we can actually see here this in this particular example with the green neuron or the green axon talking to the yellow dendrites. When we look at a axon to a cell body, this is called an axiosomatic. This is going to be where the red axon is talking to the yellow neuron's body. We then see there's the axioaxonic, which is going to be a synapsis between two axons. This is going to be where you see here with the blue axon talking to the yellow axon. All right, so it's based on what two parts of the cell are communicating. If you'll notice, the axon's always first, because that's what we're talking about from the presynaptic, but the postsynaptic could either be on the dendrites, the cell body, or its axon. Another thing to note here is there are going to be a lot more synapses than there are going to be neurons. There's going to be two basic types of synapses. Here there's another clip, and this is also again from Crash Course, which covers the different types of synapses. But we see that there's an electrical synapses. Electrical synapses is found in smooth muscle, cardiac muscle, and your central nervous system. So these are going to be between neurons. This is going to allow for a two-way communication between the cells. The ionic current can spread directly from cell to cell through gap junctions, and it stays as an electrical signal the whole time. There's no changing of the signal, so it's a very fast communication. This allows for quick coordination to take place, where the cells are going to synchronize what they're doing, which is super important when we're talking about like your heart muscles. They need to be synchronized in order to beat together to push the blood, or your bladder or something like that. Okay, so these are going to be what we see with electrical synapses. In the video from Crash Course, what they do is they compare that to like a group text. All right, all the information is going through very quickly, and it's allowing for a potential response to happen quickly. So again, the ions are going to flow in both directions. The chemical synapses is actually a little bit slower in how it communicates, but these are going to be more common. These are going to be found a lot of times on your neuromuscular junction, so between the nerve and the muscle, and this allows for one-way communication to take place. This is where the presynaptic neuron is going to talk to the postsynaptic either neuron or the effector, the muscle or the gland. So the neuron that is going to send the message and release the neurotransmitter is the presynaptic neuron. The neuron that has the receptors to receive the message from the neurotransmitter is going to be the postsynaptic neuron. Now, the presynaptic neuron a lot of times is going to be named based on what kind of neurotransmitter gets released. It could be what we call a chlorogenic neuron if it releases something like acetylcholine, or it might be an androgenic neuron if it releases like norepinephrine. Now, this neurotransmitter, this signal that gets sent, is going to be diffusing across a gap between these two neurons. This gap is known as the synaptic cleft. Okay, so the synaptic cleft is between these two neurons. So guys, the presynaptic neuron is going to release the neurotransmitter. It's going to then diffuse across that synaptic cleft, that gap, and then it's going to bind to the receptors that are found on the postsynaptic neuron. Okay, so this is going to go from electrical impulse to chemical back to electrical. Okay, so we're changing the type of signal, so this is why it takes a little bit longer. So an example here is the axio-dendritic synapses. So this is one axon talking to the dendrites on the next neuron. So we have the presynaptic neuron is going to send its action potential down to its terminal end. So this is going to be electrical. At the terminal end, it's going to release its neurotransmitter. This neurotransmitter is going to travel across the synaptic cleft, and then it's going to come in contact with the receptors that are going to be found on the dendrites of the postsynaptic neuron. If the signal is sent enough, we see that it's going to generate a graded potential for that electrical signal. Then an action potential may occur. Okay, so this is going from electrical to chemical to electrical. So the postsynaptic potential is the type of graded potential that is caused. Does the neurotransmitter actually cause a hyperpolarization or a depolarization of this postsynaptic neuron? So let's take a little bit better look when we talk about more of an animated picture with this. So step one is going to be where the action potential reaches the synaptic end bulb of the presynaptic neuron. Step two is going to be depolarization of the membrane, which causes an opening of voltage-gated calcium channels. Calcium rushes in, and this allows for the exocytosis of the neurotransmitter to take place. This exocytosis then is going to allow for the neurotransmitter to diffuse across the synaptic cleft. When it diffuses across the synaptic cleft, it can bind with the receptors on the postsynaptic neuron. Once it binds with the receptors, this is going to result in either a hyperpolarization or a depolarization of the postsynaptic neuron. It depends on what kind of channels get open. If the postsynaptic potential reaches its threshold, it triggers then an action potential to go down this new axon. Let's look at this excitatory versus inhibitory postsynaptic potential. If it's an excitatory neurotransmitter, depolarization of the postsynaptic neuron membrane will occur. This is going to be where the inner membrane becomes less negative, so cations move in or anions are going to move out. This generates what we call an excitatory postsynaptic potential, an EPSP. This causes the cell to get closer to that threshold, which is at -55. Now guys, a single EPSP is not enough to start an actual nerve impulse. However, the more that are received, the more excited and closer to the threshold we can get. Okay, so the next EPSP may be enough to start the actual nerve impulse because summation is happening here since these are graded potentials. On the other hand, there's what we call inhibitory neurotransmitters. These are going to hyperpolarize the postsynaptic neuron membrane. This is where the inner membrane becomes more negative. So this could be anions in or cations out. This generates what we call an inhibitory postsynaptic potential, an IPSP. This puts the threshold further away. So instead of sitting at -70, it could go down to like -90. This is where it's going to be harder for the nerve to end up sending an action potential. Now, if you'll notice in the picture here, guys, you'll see that there are a number of synapses talking to this particular nerve, and you'll notice that some are excitatory and some are inhibitory. This is where that summation comes in. So each presynaptic neuron may release several different neurotransmitters at the same time. Each postsynaptic neuron has its own specific receptors for these neurotransmitters. So if there's no receptor for it, then it cannot listen to that neurotransmitter. The same neurotransmitter could be excitatory in one location or area, but then it could be inhibitory in another location. It depends, all right? And so in your textbook on page 536, it talks a little bit about this. So neurotransmitters are quickly removed from the synaptic cleft, and this is going to happen a number of ways. First, it's going to be the diffusion out or away from the synaptic cleft, because remember, molecules like to move from high to low. Another thing that can happen is that enzymes can come in and degrade them, can actually break them apart and remove them. Or there could be an uptake into surrounding cells. This could be neuroglial cells, or it could be a reuptake by the neuron who sent out the neurotransmitter itself. Now, each postsynaptic neuron receives input from many presynaptic neurons at once. And so some are going to send excitatory signals and some are going to send inhibitory. This is where the sum of all the messages are going to be important. So if the excitatory are greater than the inhibitory, then it will depolarize the nerve, potentially triggering an action potential. If the inhibitory are greater than the excitatory, the postsynaptic neuron will hyperpolarize. This sends it further from the threshold, so no nerve impulse will be sent, no action potential will be sent. So let's talk a little bit about some of these neurotransmitters that are associated with the central nervous system and the peripheral nervous system. So neurotransmitters are molecules that are released as a result of nerve impulses from the axon terminal. Remember, they are going to diffuse across that synaptic cleft, and they are going to potentially talk to the postsynaptic neuron as long as the postsynaptic neuron has a receptor for it. One example is acetylcholine. We talked a little bit about acetylcholine back in the muscle chapter. It's going to be released by cholinergic neurons, and this is going to be both in the somatic and the autonomic nervous system. It is excitatory whenever acetylcholine is sent to a skeletal muscle and it causes the skeletal muscle to start contracting, but it is inhibitory if it's sent to the heart muscle, the cardiac muscle. This actually causes your heart rate to slow down. Amino acids are another type of neurotransmitter. We have the excitatory in the central nervous system as glutamate and aspartate. In the central nervous system, some inhibitory amino acids are GABA, and this is found in the brain, and glycine, which is in the spinal. Biogenic amines can be excitatory and or inhibitory based on the type of receptor at the end of the synapses. In general, these are going to be responsible for your sleep-wake patterns, your feelings, your moods, and your emotions. Some examples are the norepinephrine and epinephrine. So adrenaline is another name for epinephrine, or noradrenaline is for norepinephrine. Norepinephrine, epinephrine, dopamine, and serotonin are all going to be what we consider anergenic neurons. They're going to be sent out by the sympathetic subdivisions of your autonomic nervous system. These are what we call catecholamines. They are going to be synthesized from the amino acid tyrosine. And an imbalance of norepinephrine, dopamine, and serotonin are actually linked to depression and bipolar disorder. Some other examples are purines like ATP, ADP, or AMP, nitric oxide, carbon monoxide, and neuropeptides are also known as endorphins. These can either be excitatory or inhibitory depending on their location. Now, we do know that there are some disorders that are linked to neurotransmitters. This includes like Parkinson's and schizophrenia. We know these are linked to the neurotransmitter dopamine. Alzheimer's, there is a link to the acetylcholine neurotransmitter. Depression, bipolar, or what we call seasonal affective disorder, or SAD, is linked to serotonin, norepinephrine, and dopamine. This is why certain drugs like Prozac, Paxil, or Zoloft are going to inhibit the serotonin reuptake. This allows for the serotonin to remain in the synapses longer to stimulate those cells to help keep you happier. It tries to stabilize the mood. Treatments for a faulty neurotransmitter might be a drug that is going to mimic the neurotransmitter, or it might block or inhibit some sort of transmission of the neurotransmitter. Some factors that can modify the effects of a neurotransmitter are their synthesis. Is it stimulated or inhibited? In Parkinson's, it's actually inhibited. It decreases the synthesis of dopamine. The release, is the release enhanced or blocked? When we talk about amphetamines, they're going to enhance the release of dopamine and norepinephrine, but botulism toxin is going to block the release of acetylcholine. So it depends on which kind of stimulus we're wanting. Receptor sites could be activated or blocked. This is like Cruar was going to block the acetylcholine receptors in a neuromuscular junction. This causes muscle paralysis. Or neurotransmitters could get removed from the cleft. Either they could stimulate the removal or inhibit the removal, which is what we see with cocaine. Cocaine inhibits dopamine reuptake, which causes a euphoria to take place. It's going to excessively stimulate the brain, so then the brain's going to get rid of a lot of those receptors, so it does not get overstimulated. Other factors that can modify the membrane potential is the increased excitability, reducing the threshold. It makes it easier to depolarize it. And we see that alkalosis can do this. It causes the patient to be lightheaded, tingling or numbness spasms to take place. But also caffeine and nicotine can also cause your nerves to be more excitable. Decreased excitability is going to increase the threshold and makes it harder to depolarize. This is seen in acidosis. It causes the patient to be weak, lethargic, and even go into a coma. But we see hypnotics, tranquilizers, and anesthetics are used to decrease the excitability so that these neurons do not send their signals. The last thing in here is the neural circuits. When we look at neural circuits, guys, one of the last things in these notes is neural circuits. This is where the neurons in the central nervous system are organized into circuits, millions of neurons, and even more synapses. I want you to just know that there's several different types of circuits. You can see them located here. Some of them are going to diverge starting in one spot and spread out. Others are going to spread out and converge into one area. There's a lot of different types. But guys, epilepsy is when these circuits with these neurons in the central nervous system are abnormal. So they start to short circuit and do not send signals like they should. Now, regeneration of nervous tissue. Guys, at approximately 6 months of age, neural cell bodies lose their ability to do mitosis. So they can't divide anymore and we can't replace them. However, in the peripheral nervous system, the myelinated axons and dendrites could be repaired if the cell body and the Schwann cells are not damaged. They can actually work on repairing those. These could also be surgically reattached if a nerve is severed, as long as the actual cell body is not damaged. In the central nervous system, however, injury cannot be repaired. So learning does frequently stimulate production of new neurons in what we call the hippocampus of the brain, but no repair of injured neurons can take place. The ability of your brain to reroute, or to actually be able to, when you learn, be able to make new connections or new circuits, is called plasticity. It's the capability of your brain to change based on experience and learning.